Hey everyone, we're going over the cell right now. Here we have a cell model, and we have another cell model right here, which we'll compare with, and then two or three models showing specifically just the membrane. Let's take a look at the cell model first. So we'll start with the most identifiable feature of any cell, and that's the nucleus. The nucleus, all nucleus means is the center of something. There's a nucleus of an atom. We'll talk about nuclei in the nervous system. So this is the nucleus of the cell. It is made of membrane, so this is a membranous outline structure, and it contains DNA. It's there to protect and regulate DNA. DNA are instructions. So on these DNA molecules are a set of nucleotides, and that gives you instructions on how to make something. We'll get to what it makes in a second. So this DNA should always stay protected in the nucleus. We never want the DNA to leave. But it has instructions. So let's say you're at the library and you want to get instructions from a book, but you're not allowed to check the book out. What should you do? You write it down on a new sheet of paper. You transcribe it. So DNA is transcribed into a molecule called mRNA, messenger RNA, and that can leave the nucleus. You see all these pores here, these nuclear pores? The mRNA can leave the nucleus, and it makes its way to these brown bumpy structures. These are ribosomes. Ribosomes are a mix of protein and nucleic acid, but ribosomes are a type of organelle, not membranous, and they take that mRNA and they read the transcript. They read the transcript and they take special building blocks called amino acids and put them together to form a new molecule called proteins. That process is called translation. So we've gone from DNA, which has instructions, mRNA, which has a transcript of the instructions, and then we're using those instructions at the ribosomes to make protein. That's translation. Now that we've got this made protein, it's not actually in its full form yet. We need to, let's say we're making a box, but we just have the cardboard, we need to fold it into its three-dimensional functional shape. So this protein is not three-dimensional yet. We need to make it functional. It's just this one long strand. That protein will get fed into this organelle that these ribosomes are on top of. When you see this membranous organelle, membranous like the nucleus, this membranous organelle with the ribosomes attached to it, this is called the rough endoplasmic reticulum. You can see that here, and here, and here, and here, and here. The rough endoplasmic reticulum is important for processing proteins. So we're properly folding it into protein's three-dimensional functional shape. Once that's done, the protein will then journey to the next place. The next place is called the Golgi Apparatus, or Golgi body, or Golgi complex, the Golgi something. We send the protein within vesicles. A vesicle is a pinched off piece of membrane. That pinched off membrane can float and connect and fuse together with the Golgi apparatus. Within the Golgi, think of this as your postal center. We need to figure out where this protein is going to go. So is this protein going to go somewhere else in the cell where we need it? Is it going to go somewhere embedded in the membrane? Or are we going to have it exocytose and leave the cell? The Golgi is needed for sorting and shipping protein to the correct spot. So let's say this protein gets packed into vesicles, here are vesicles, and we're going to ship them out and have them leave the cell by exocytosis. So here the vesicle meets with the plasma membrane and fuses together, because this is all membrane. Membrane, 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 membrane. It's all the same material. And so this vesicle can fuse with the plasma membrane and release anything to the outside. Or if that protein is embedded, it then gets incorporated with the plasma membrane itself. All of that has to do with protein production and release of protein to different places. The nucleus, ribosomes, rough endoplasmic reticulum, Golgi apparatus, plasma membrane. Other organelles include this one that looks just like the rough endoplasmic reticulum, but it does not have ribosomes. Since it doesn't have ribosomes, it has nothing to do with protein production or regulation. These are the smooth endoplasmic reticulum. Smooth because there are no ribosomes. Smooth ER is good for detoxifying harmful substances like alcohol, so you'd find it a lot in your hepatocytes, your cells of the liver. It's also good for processing lipids, so if you want to assemble certain steroid hormones like testosterone or process any fats from your diet, that can be done here in the smooth endoplasmic reticulum. These organelles shown here, these are mitochondria. Mitochondria are the powerhouse of the cell. They have a highly folded inner membrane. What does that mean to be a powerhouse of the cell? When we eat food, one major molecule that we use for energy, I guess the three major molecules we use for energy are carbohydrates, like sugars, proteins, and lipids. All those molecules we can break down and use for energy. But carbohydrates and lipids and proteins themselves are not in a form that our cells can use. Think of it going to another country with your American dollars. You can't spend those American dollars in another country. You have to convert it first to a different currency, and then you can spend it. Glucose is a different currency. We need to convert it to the currency of the cell, and that's called ATP. It stands for adenosine triphosphate. We'll talk about that later. Mitochondria are important for the chemical reactions for converting things like glucose into ATP, the energy from glucose, and storing that in ATP. So we have this really highly folded inner membrane that by being highly folded, we're packing more membrane into a smaller space, and we can more efficiently produce more ATP. There are several other structures that look just like vesicles, like this one here or this one here. I honestly can't tell you which one's which, but any vesicular structure that does more than just transport, let's say this vesicle contains enzymes that help break things down, digestive enzymes, this would be a lysosome. The word lys means to break down, break apart. So a lysosome is an organelle that would break things apart. Let's say this is a white blood cell, and this white blood cell ate something up. We bring a vesicle in. This lysosome can fuse with that vesicle, release its enzymes, and digest that thing. So lysosome is good for digesting foreign things like bacteria or old organelles. Say this mitochondria is old and doesn't work anymore. We can break this down and use it for scraps. There's going to be useful molecules that we can use from it. Another vesicular organelle is a peroxisome. Peroxisomes contain peroxide or something similar to it and help detoxify certain things. So more chemical reactions go in there. So the ones that look like vesicles are vesicles themselves, lysosomes, and peroxisomes. Besides that, you can find cytoskeleton, and actually these are centrioles, which are used in mitosis. Centrioles have microtubules that allow for the attachment to chromosomes and splitting that, which we'll talk about in a different video. The fluid of the cell is called the cytosol. Cytosol literally means solution of the cell, or you can call it intracellular fluid. It's the same, it means inside of the cell. And in the cytosol, that's where you find the organelles and all kinds of nutrients and ions and waste all dissolved in the liquid. And the outside is the plasma membrane. That's made of phospholipids. It's a phospholipid bilayer. So this model is showing what a single phospholipid roughly looks like. It's got a head that's hydrophobic, a head that's hydrophilic, which means it's attracted to water, and tails that are hydrophobic. So they're scared of water, which is why the tails are facing each other. They don't want to face the watery interstitial fluid or the inside of the cell, which is also watery. So the hydrophobic tails are protected, and because of these hydrophobic tails, that makes the membrane, makes a barrier. We have a hydrophobic barrier. So things that dissolve well in water, like glucose or ions, they can't easily penetrate through the membrane. We still need to get things like glucose or ions into or out of the cell, so we need a way for them to transport in and out of the cell. And that's what proteins embedded in the membrane are important for. Shown in this model, these styrofoam heads are the heads of the phospholipids, here are the tails, and you can see the bilayer here. There's two layers of phospholipids. We need proteins that span that bilayer to allow things to pass back and forth. Those are generally known as transport proteins. There's a million other types of proteins found in the membranes, like anchoring proteins or receptor proteins that can also be found in the membrane. But let's say that this one's a transport one, since it has this passageway that can allow things to pass in and out. Besides that, in the plasma membrane, you can find things like cholesterol. Cholesterol, I know we think of as a bad molecule, but we need it for a bunch of things, including producing hormones like testosterone and estrogen, but also the cholesterol can be found in membranes, and it helps keep the membrane more rigid, more secure. Think of the membrane as like the surface of the ocean. If you have a surface of the ocean that's pretty wavy, if you shake it too hard, you'll break that wave, and we don't want our membrane to break. The cholesterol helps keep that wavy membrane more rigid, so it's there interspersed for support. On the surface, on the outside of the cell, you'll find these carbohydrates. It's known as the glycocalyx. These are glycoproteins or even glycolipids. They're for sensing what's on the outside of the cell, for for our own body recognizing what these are, or even just for stickness. So there's a number of things that these could do. Just to point out some differences in this model. In this model, we can see ribosomes shown here as white dots. So this is rough endoplasmic reticulum. Here are mitochondria. If we find one of these without dots, like, actually I'm having trouble finding one. What do they say is the Golgi? It's 5, and then is there any smooth ER? No smooth ER. All right. So I don't find any smooth ER, but we have the Golgi here, and you can see vesicles breaking off. These are centrioles, things used for mitosis. This yellow one and this brown one of them is a peroxisome, one is a lysosome. Yeah, that's what we find in the cell.